Hi there, welcome to AP Biopharma with Dr. D, and today we're going to be talking about regulation of gene expression in eukaryotes. Okay, so I would like to remind you that both prokaryotes and eukaryotes can alter gene expression in response to their changing environment. And the connection here is to topic 5.5, where we talk about environmental effects on phenotypes, if you remember. Now, in addition to responding to their environment, multicellular eukaryotes, such as humans, also develop and maintain multiple cell types. So, and this is the result of gene expression. Gene expression is often regulated at the transcription stage, but control at other stages is important too. So, random questions. So, we all started as a single cell that fertilized egg. That cell multiplied, divided into a trillion cells by the process of mitosis. And if mitosis makes an exact copy of itself, it's a form of cloning, how come every cell in your body isn't exactly the same, right? How come they all have different shapes, sizes, and different functions? Well, that is the result of differential gene expression or differences in gene expression. Despite having the same DNA, different cells in our bodies express different genes. And this is the basis for cell specialization, cell differentiation. Eukaryotes can regulate gene expression in both a spatial and a temporal manner. And what do I mean by spatial? Well, different cell types and different tissues occupy different space. So different genes are expressed. And during early embryonic development, depending on where cells are, depending on their location, that will determine what type of genes are to be expressed, and that will ultimately determine what cell types they will become. Temporal expression is when different genes are expressed at different times of an organism's life. For instance, early development versus adult, or different genes can be expressed at different times of a cell's life. So they're expressed only when they're needed. And here is a great illustration of how the body plan of animals, including humans, is determined by differential gene expression. And what you will see here is that the different genes shown here are expressed in the different parts of the developing embryo, both human or fruit fly. And depending on where they're expressed, that will determine what kind of body parts will develop: head, tail, wings, and so on. Now, here's some examples of temporal regulation. Genes responsible for rapid growth and development are on during early development and are being turned off in adults because we as adults do not have many cell divisions. So we don't need those genes. Reactivation of those genes can lead to cancer. And an example of that we've already talked about when we talk about DNA replication is the enzyme telomerase. Genes which are required for DNA replication are also regulated in a temporal manner. They're turned on only when they're needed in late G1 and early S phase, and they're not expressed in non-dividing cells. A non-dividing cell does not need any of the enzymes involved in DNA replication, so these genes will not be expressed in a differentiated cell that is not dividing. In all organisms, a common control point for gene expression is at transcription, the level of transcription. However, the greater complexity of a eukaryotic cell structure and function provides opportunities for regulating gene expression at many additional stages. So this is an overview of all the stages at which gene expression can be regulated. It's being regulated at the chromatin level, at the level of transcription, RNA processing, transport of the mRNA into the cytoplasm, translation, protein processing, and protein degradation. So what is the purpose of all these regulatory steps? Why do we have this many? Well, it really allows for a greater control. So we don't just have on and off. We can have anything in between. And because of that, we have the ability to respond to a greater variety of signals. So regulation of gene expression, what does it mean? Well, the majority really occurs at this level, at the level of transcription. And in this model, I am showing you the basic structure of a gene, okay? So what you will see here in red, that red arrow, is really what the gene is. There is this thing here called +1, which essentially is the first nucleotide that is going in DNA that's going to be read and turned to an RNA. And from here to here are the DNA sequences that encode the information for RNA and that will be turned eventually into a protein. To the left of this, we have DNA sequences that do not code for proteins or for mRNA. They are regulatory sequences. Just like the operator and the promoter in bacteria, these sequences are regulatory sequences. They regulate transcription. So in order to understand how transcription is controlled in eukaryotes, we need to know that it involves two components. One, the portions of DNA, which are the regulatory regions that I just showed you. They're switches. They're part of DNA that do not code for proteins, but are responsible for controlling mRNA production. And two, they're actually proteins that bind to these regulatory sequences. They're called transcription factors, and they flip those switches. They are used to regulate the production of RNA. So regulatory DNA region. By definition, the transcription start site, or +1, is the first nucleotide that will be added. We also have core promoter elements. So let me show them again. So these are core promoter elements. So this is the regulatory sequences which are immediately adjacent to +1, to the start of transcription. These core promoter elements are common to most genes, okay? They are the place where transcription factors will recruit RNA polymerase and where RNA polymerase will bind. An example of such common element is the TATA box. It's an AT-rich region, which is easy to melt. And of course, there's gene-specific promoters or gene-specific regulatory sequences. They're binding site for regulatory factors that are specific for specific genes. They do not regulate every gene. They regulate only a certain set of genes. Some of them are quite close by. They're located about 100 nucleotides from +1, and some of them can be very, very far away, and they're called enhancers. They could be located tens of thousands of nucleotides away from the start site. Okay, so what are the common ones? Well, the common promoter elements, the common regulatory elements bind to these general transcription factors. And they're called general because they're common to all genes. All genes require them. And usually they're referred as to pre-initiation complex. Why? Because it contains RNA polymerase, which is necessary, and it contains a bunch of proteins. There's like tons of proteins here whose job is to help RNA polymerase bind to the right site and help it initiate transcription at exactly the right place. Okay, they are also responsible for melting the DNA helix to expose the bases so that RNA polymerase can now start synthesizing RNA. Gene-specific transcription factors are specific to specific sets of genes, so they're not common. They can either activate or inhibit transcription, so they're called either activators or repressors. They can bind close to RNA polymerase, or they can bind very far away to enhancers. And they have two main jobs. One is to aid, if they're activators, or inhibit, if they're repressors, the recruitment and the assembly of the down transcription factors, and therefore RNA polymerase. Two is to respond to regulatory signals. So these gene-specific transcription factors are actually responding to signals. Remember when we talked about in Unit 4 how cellular signaling eventually leads to changes in gene expression? So these transcription factors are the ones that are actually responding to those cellular signals and will either turn on or turn off the expression of specific genes. So that's one of their jobs. So how do they affect transcription? They could do that in both a positive or negative way, right? Positive if they're activators, negative if they're repressors. Well, they can directly interact with RNA polymerase and with the transcription factors and help recruit it, help stabilize them. Or they can do that indirectly by affecting the structure of chromosomes, by affecting chromatin structure. And we're going to talk a little bit about this later, but chromatin, the assembly of DNA into chromatin, actually represents a barrier to transcription. And these transcription factors can affect the structure of chromatin and therefore influence indirectly transcription. So activation versus repression. In eukaryotes, always we have both. We have both repression and activation at a single gene. And why is that? Well, it allows us more control. We can regulate the regulators. It also allows us more control in terms of how much RNA is made. In eukaryotes, it's not all or nothing, like in bacteria. You can have a lot of RNA. You can have intermediate amount of RNA. You can have very little RNA, or you can have no RNA made whatsoever. So there is a gradation of how much RNA you make and how much protein you produce. Genes in eukaryotes are regulated coordinately. So unlike the genes of prokaryotic operon, each gene, so when genes are actually, when you need to turn on multiple genes at the same time, so for instance, you need to turn on all the genes that code for all the DNA replication proteins, they need to be all turned on at the same time. They're not organized like in bacteria and operon. They simply have common regulatory elements. They have common control elements, which means same set of transcription activators are going to bind to them and are going to turn them on at the same time. So the genes can be scattered over the chromosomes, but each gene has the same combination of control elements, which means the same combination of transcription factors is going to bind to them and turn them on at the same time. Now, the other thing that we need to understand is that what makes a gene being expressed in one type of cell and not another really is the presence of both regulatory elements and the transcription factors that bind to them. Okay, and we're going to do a very good activity to help us understand this. But here's an example of two genes. One is the albumin gene, and the other one is the crystalline gene. The crystalline gene is expressed only in the lens cells of our eyes. Albumin gene is only expressed in the liver cells. They have a core promoter element. That means all genes have the same promoter. And they have enhancer, gene-specific regulatory elements, which are different. So what will happen here is that one of the transcription factors here, which is in gray, can bind to both genes because they have that regulatory element. However, the yellow and the red are only present in the albumin gene, and the orange and pink are only present in the crystalline gene. So there's one common transcription factor and one common regulatory element, but the other two are unique. So in a liver cell, only the albumin gene will be expressed, but not the crystalline gene. Because in a liver cell, we only have the red and yellow transcription factors. We do not have the pink or the tan or orange transcription factors. Conversely, in the lens cell, the crystalline gene will be expressed because we have the pink and the orange transcription factors to bind to those enhancer elements, but we do not have the red transcription factor or the yellow transcription factor to bind to the albumin gene. So the albumin gene is not turned on, okay? Now, here are the key points to remember. Transcription involves a large transcriptional machinery. There's 50 proteins just for basic regular transcription. RNA polymerase and all of its accessory proteins, which are called the basal or the general transcription factor. The main control is at initiation. Do we turn the gene on or do we not turn it on? Assembly of DNA into chromatin represents a major barrier to transcription. And the job of transcription activators is to help the basic transcription machinery to gain and retain access to promoters. Whereas the job of repressors is to prevent that, to hinder that access to promoters. Transcription activators and repressors are also key transducers of regulatory signals. They're key effectors of regulatory signals. So we're gonna talk more about chromatin, but right now we want to talk about another step in regulating gene expression, and that will be RNA processing and export of the mature mRNA out of the nucleus. Because transcription and translation are compartmentalized, one occurs in the nucleus, the other one occurs in the cytoplasm, that gives eukaryotic cells the opportunity to regulate. So proper RNA processing is really necessary for nuclear export. So we're going to cover processing and mRNA transport. And we already know that the 3 steps in processing are adding a 5' cap, and the cap is required for initiation of translation, adding a 3' poly A tail, which determines the stability of the mRNA, and splicing, removal of introns, right? And I would also remind you that we can regulate gene expression by regulating alternative splicing. So once in the mRNA, once in the cytosol, the mRNA can be and will be eventually degraded. How long an mRNA lives really will determine how much protein you can make. Once the mRNA is translated, the protein can also be modified. It can be either modified by adding certain covalent modifications, or it can actually even be degraded. So these are other steps in the regulation of gene expression. So mRNA degradation. mRNA degradation is regulated in large part by sequences at the 3' end of the molecule. And they're called the untranslated regions of the mRNA, or the 3' UTR and the poly A tail. And I would like to point out to you that this is where transcription starts of the gene, and this is where transcription ends. But only part of the mRNA is actually going to make it into a protein. That is the coding region. So the first codon in translation, AUG, is not always, it's actually rarely the first nucleotide that's being transcribed. There is a region at this end that is not being translated. It's called the 5' UTR, untranslated region. And there's a region at the other end, right after the stop codon for translation, that is also not going to make it into the protein. It's not being translated. And these 2 regions regulate mRNA stability and translation rates. So they're regulatory regions. So the rate of translation is regulated. The rate of translation will depend on A, how long the mRNA lives, and B, how many ribosomes are going to cover that mRNA, how quickly are they going to be translating it. So translation can be regulated like all or nothing, or you can have like really fast translation and really slow translation. So there's also in between. And again, the sequences responsible for regulating are these untranslated regions. The part of the mRNA that is before the start codon, the 5' UTR, and the part of the mRNA that is after the stop codon, the 3' UTR. They don't really carry information for building the protein. They're regulatory, okay? And they're different for different mRNA molecules. Proteins that can bind to these regions can regulate the rate of translation. In addition to this, translation can be regulated by alternatively, translation of all mRNAs in a cell can be regulated at the same time, simultaneously. So translation initiation factors, for instance, are responsible for activating translation of all mRNAs in an egg following fertilization. So post-translation, what happens post-translation? Well, once the protein is translated, sometimes it is produced in a non-functional state, so you need to modify it. And we've already encountered some of those modifications. They're called phosphorylation, so adding phosphate functional groups to a protein. And those can alter the shape of the protein and make it functional. Sometimes proteins can also be glycosylated, so one adds sugar to them, it becomes a glycoprotein. And often proteins, after they're being produced, are being degraded. They're being shot out into a special machinery called the proteasome, which will chew up the protein and recycle the amino acids, and thereby effectively shutting off gene expression. Because remember, gene expression is the production of a functional protein. So when you remove the protein, you no longer have expressed that gene. Okay, I am going to stop right here, and we're going to continue with this next time. And we're going to start talking about how chromatin and the packaging of DNA influence gene expression.